Our next speaker is Evan Eskew. He's a PhD student here at UC Davis in the graduate group in ecology. He's got his undergraduate degree from Davidson College in Davidson, North Carolina, and where he worked on applied reptile and amphibian conservation. And he's now at UC Davis in the lab of Dr. Brian Todd, working on the amphibian disease, chytrid fungus. So Evan's talk is called The Effects of River Flow Regulation on Anuran Occupancy and Abundance in Riparian Zones. Welcome, Evan.
All right, thank you everybody. Thanks for sticking around to the bitter end with me. I'll go ahead and say that I feel like a really big imposter up here. If I'm anything, I'm a herpetologist interested in reptiles and amphibians. So I got interested in freshwater science or aquatic biology through that realm. Also, a geographic imposter. I am not going to tell you about work from California. This is stuff I did in the East Coast, in North Carolina and South Carolina. So, to start out with, again, sort of preaching to the choir, damming is a pervasive impact on river systems worldwide. The last estimates I saw were that there are about 800,000 dams in the world. And actually, as I was preparing for this talk, I found this article on the front page of NBC News, actually, which was reporting the planned development of 3,700 new large hydroelectric dams. The interesting thing about these was that they're primarily in developing parts of the world. You know, many developed countries have already built most of the dams that they're going to. And so this new development that this article described would dam, I think it was 21% of the largest 120 free-flowing river systems in the world, if all those go as planned. So damming is an issue that we need to understand now, and it doesn't look like it's slowing down anytime in the future. So obviously the ecological impact of dams are going to vary greatly depending on the size and the type of dam on the river system, from something like the small mill dam to something like the Three Gorges Dam in China. So this is the largest concrete structure in the world, and a very different effect on the river system, on the ecological community around this impact. But there are some general ways that we think that dams would affect riparian and riverine organisms. One of those is pretty obvious, is just a lack of dispersal or dispersal limitation. So dams provide a physical barrier to dispersal for many strictly aquatic species, like the sturgeon, unless we specifically design a dam in order to mitigate this impact. Another one is, unsurprisingly, dams modify downstream flows. That's why they're built in most cases. And the effect of dams is typically to stabilize downstream flows. So the lowest flows become, the low flows become higher, and the peak flows become lower. And in addition, dams are going to control flooding events. So alteration in the scale of those flooding events can change the hydro period of riparian zone wetlands, or the distribution pattern of those wetlands. And then finally, in some of the larger dams, where the water releases from the dams are controlled by us in order to generate power at appropriate times, these water releases can be very high flows and very sudden. And so depending on when those happen, you can actually physically displace organisms in the river channel and scour either eggs, larvae, or adult organisms. So the ecological impacts of dams have mainly been studied in strictly aquatic fauna. So you have your macroinvertebrates, mussels, and fish. And what many studies find is that there are effects on both diversity, so species richness, and abundance, which are most severe, as you might expect, immediately downstream from a dam, and those impacts lessen as you move further downstream. The flow regime becomes more natural. So here's an example of that. What you have, let me find this again. So you have the number of mussel species on the y-axis. Immediately downstream from the disturbance, you have relatively low diversity. As you move further downstream from that disturbance, that picks back up and increases to a more natural level. Now we know that Anurans, so frogs and toads, are important members of riparian zone communities, but compared to those strictly aquatic species, we don't know a lot about how damming influences their populations. And there's a few reasons to think that it may be different than strictly aquatic species. For one, as you might be familiar with, many Anurans have this sort of classic biphasic lifestyle. So they start out, well, after they hatch from an egg, they start out as a strictly aquatic tadpole stage or larval stage in the salamander. Then they metamorphose into a more semi-aquatic adult stage. And so obviously there's reason to think these two different life stages may be differentially affected by the impacts of damming. In addition, there's a wide variety of variation in life history or natural history among Anuran species. So different species breed at different times of year, and they use different sorts of habitats for that reproduction. So things ranging from a free-flowing river, from a main stem of a river channel, to seasonal wetlands, or even more permanent bodies of water like this. And so depending on which of those habitats they prefer, dams may affect them differently. In addition, we know that, like many other species, urbanization has a serious impact on the Anuran populations. So, and urbanization can also interact with riparian zone impacts through damming, both directly. So urbanization could occur in a riparian zone where you then lose that riparian habitat, or it could contribute more indirectly through runoff, things like that, from urban areas into a riparian zone where the organisms there are then negatively affected. So the goal of this work was to, one, evaluate the effect of dam proximity on occupancy and abundance of Anurans inhabiting riparian zones. And so occupancy is just the presence or absence of species, abundance, obviously, their number. So these are really just two different ways to evaluate, or two different population metrics to use when studying Anurans. And then we also wanted to evaluate the effect of urbanization to see how that compared with the effects of damming, or how it might interact with the effects of damming. So this work was all conducted in South Carolina, so on the Broad and Packlet rivers, which extends over most of the upstate of South Carolina. So the inset here, shows our study region, the Packlet and the Broad rivers. Everything in red are 42 study sites, and the 16 green crosses are the dams that we identified along these river reaches that we studied. These dams are a wide variety of uses, so things from historic mill dams up to big hydroelectric generating facilities. To gather information on the distribution of Anurans in this study region, we conducted manual calling surveys, which is a science way of saying we listen for frogs. So this is a really convenient thing about studying Anurans. They have species-specific calls, so you can just go out there and listen to them. So some of the best field work you'll ever do, you can dress in your sandals, you can drink a lot of Red Bull and eat a lot of gas station food and drive around at night and listen for these organisms. And so we did this often. So we conducted surveys in 3 different calling windows at 3 times of year, spring, summer, and winter. And that's in order that we would detect species that breed at these different times of year. And then we also conducted 3 surveys within each of those calling windows. So in total, we had 9 surveys at each of our 42 sites, stretched over this roughly year-long time span. This is an ugly slide, but stick with me, it's somewhat important. So then once we had this data, this presence-absence data, I also forgot to mention, with the calling survey, you not only can detect a species, obviously, if you hear calling, but you can also rank it to some degree on the scale of abundance. So do you hear one spring peeper calling, or do you hear a whole chorus of them? And so that's like sort of a semi-quantitative method. But then to analyze our data, we actually use program presence. And so the really important thing about that is that it allows us to estimate occupancy and abundance of these species while accounting for imperfect detection. So, for instance, there could be a site where you go out and you listen 9 times and you never hear a Fowler's toad, but Fowler's toads were actually there. You just sampled at the wrong time, they didn't feel like calling that night, whatever the case may be. And so what presence, any sort of occupancy kind of framework allows you to do is to account for that imperfect detection and get a better estimate of the real distribution of your species of interest. And so the numbers here aren't important, but what they do illustrate is that for each species, for both occupancy and abundance, we have this model set. What these model sets allow us to do is to explicitly test our hypotheses. The way we did that is we had covariates for each study site. So the covariates that we measured were the distance upstream and downstream to a nearest dam. So that gives us an idea of the impact of damming. And then also and all this was in RJS, sorry. Then we also measured the urbanization, the impervious surface, buildings, roads, in a buffer zone around each study site. So we have these three different covariates, upstream to dam, downstream to dam, and urbanization for each study site. And so by incorporating those into our models, if the models are highly ranked, indicating strong support for that model, and it contains a particular covariate, then we have good inference that that covariate influences the population metrics that we're seeing. So, to get to our results, we actually detected a total of 13 species over that year-long sampling, 9 of which had data that was sufficient to analyze with presence. So those are listed here, representative pictures at the bottom, including a calling Fowler's toad. Notice we didn't use all 9 surveys for each species, and that's simply because these species don't breed year-round. So it doesn't really make sense to include surveys for a species at a time of year when you would never expect it to be calling. So our actual number of surveys used was a subset of those total based on when we would expect their breeding activities to take place. And now I'm going to walk you through one example for one species about how we sort of made our inferences. So here's a model set. So this is the occupancy model set for cricket frogs, just for cricket frogs. And what each line here, it represents a different model. And what's really important is this top model. So the top model is the one that's highest ranked in terms of model support. And what you see here is that this little side parameter, that little trident looking thing, in this model, that is influenced by the distance a site was downstream from dam. So that covariate turned out to be really important in the case of this species. The second parameter is detection, which we're not really as interested in. But what you see here is that for this species, we have strong evidence that that distance downstream a site occurred from dam influenced this species' occupancy. Similarly, for the abundance model set for this species, distance downstream from dam influences this lambda parameter, which is representative of abundance. Sorry, I think I mentioned that, but there's an occupancy and abundance model set for each species. And so essentially what we did is go through our models. We looked to see if the top ranked models for each species included an effect of a covariate, and then also whether or not that covariate was significant. So just because down from dam is really important for abundance, presence doesn't always estimate it very precisely. So you could have a significant effect here, but when you look at the parameter, you can't really tell, well, was it a positive effect or a negative effect? So all the graphs you're going to see from here on out are cases where the top model had a covariate included, and that covariate had a significant directional effect on either occupancy or abundance. But I think that'll be clear from just looking at the pictures. And so here those are. So for the occupancy model sets, what we found was 2 species, cricket frogs and southern leopard frogs, had a significant effect of distance downstream from dam on their occupancy. And in both cases it was positive, which is what you'd expect based on previous results in strictly aquatic species. As you move further downstream from a dam, a site is more likely to have either of these or both of these species. For bullfrogs, we actually found that urbanization was the most significant predictor of their distribution. Again, the trend you would expect. As you have more urban cover in that buffer zone, a site is less likely to host bullfrogs. For abundance, kind of similar patterns. Cricket frogs, spring peepers, strongly influenced by distance downstream from dam in the direction you would expect. Move further downstream, you get more of these species. The same thing for these 3 species. So abundance, we actually had a number of species that showed these strong impacts of downstream from dam. So upland chorus frogs, green frogs, and southern leopard frogs, all in that positive direction. There was one outlier who didn't behave. Fowler's toads actually were significantly influenced by distance downstream from dam, but in the opposite direction, where if you move further downstream from the disturbance, you actually got fewer of them. And for bullfrogs, again, urban cover was there, the most significant predictor of their relative abundance, again, in a negative direction. So in summary, what we found was that the downstream distance impacts appear to be greater than upstream distance, and that as we expected, those downstream distances were generally positively correlated with the population metrics we looked at. So you further downstream, more likely to contain species and more likely to have more of those species. Urbanization, kind of surprisingly, since it's so well studied and kind of obvious in its impact, it did not rank highly compared to these damming effects for many species except for bullfrogs, which was a little bit unexpected. Now, we didn't explicitly state the mechanisms of how this pattern was generated, so Hiromi did all these really awesome experiments. We didn't really do much of that. This is just sort of the pattern that we found over this year, but we can kind of speculate based on other studies about what might have been going on. So first are these reduced flows and flooding events downstream, which have the potential to reduce riparian wetland habitat that many of these species need. In addition, the flow regime changes in time. So in particular, we know that some of these, downstream from some of these dams, there are very low flows in April and May, which is right when our summer breeding species would be gearing up. So even if it's the same amount of water, the timing can be really important for these species that need it at particular breeding seasons. And then finally, downstream from some of our larger dams in the study, water release events could have been physically displacing either eggs or larvae or adults, actually. And so the implications of this work, I think, are that riparian zone fauna are affected by damming too, and that the effect of a dam can extend to those organisms inhabiting that region. It also suggests that dam removal or other mitigation strategies could potentially have a positive impact on conservation of riparian zone organisms, in addition to those inhabiting the actual river channel. And I think there does need to be kind of more work on exactly how that happens. And there are a number of research groups here on the West Coast where this has been studied in a little bit more depth than some West Coast species affected by damming. If you want to read more of the gory details, the project is in print. Some of my acknowledgements, my co-authors, and many people who helped in the field, and I'd like to thank you for your time.
Got it. Minute for questions. Any questions for Evan about his research?
Yeah.
Can you wait for microphone, please? Thank you.
For your one frog that showed a greater propensity to be near the dams, were you able to tell if there were some factors such as preference for breeding in stiller water, and your calling was actually picking up frogs that were around the lake habitat itself and not really associated with the downstream river disturbance?
Are you talking about Fowler's toads, the ones
Yes.
that showed the opposite trend?
Yes.
Yeah, so Fowler's toads did show the opposite trend. They were kind of a weird case in our data. So their naive occupancy was 95%. We detected them at all but 2 sites in our study. So they are extremely common in almost every habitat. And I think I have this, oh, I do. So if you see the 2 sites where they did not occur were immediately upstream from dams. So I think it's just sort of an artifact of our particular data set. I don't really think there's any significant biological reason to think that they do better immediately downstream from a dam. So I don't really think that that's, it's just kind of a weird thing.